And our next speaker is going to be Mona ElSisi from Ain Shams University in Egypt, who will be discussing the potential of HOTTIP SNPs as invasive diagnostic and prognostic biomarkers for HCC metastasis and siRNAs as a potential avenue for targeted therapy. Hi, Mona, and thank you for being here today. We look forward to hearing your presentation.
Thank you. Is my voice clear?
Yes, we can hear you.
Okay, thank you. I will share my presentation. Is the screen okay?
Yes, we can see it.
Okay. Starting. Good morning and good afternoon, dear colleagues. My name is Mona ElSisi, PhD candidate and assistant lecturer at Biochemistry and Molecular Biology Department, Faculty of Pharmacy, Ain Shams University, Egypt. Today, I'm excited to be part of that huge scientific conference to present you the presentation talking about my PhD thesis results. This presentation is entitled The Potential of HOTTIP SNPs as Non-Invasive Diagnostic Prognostic Biomarkers for HCC Metastasis and a Design Short Interfering RNAs as Promising Avenue for Targeted Therapy. It was a research initiative focused on studying the Egyptian cancer epigenetics in general, and in particular, the role of different variants of HOTTIP in combating metastasis. I was honored by the supervision of Professor Dr. Nadia Hamd Al-Hifni, Professor Dr. Sameh Saad Ali, Dr. Mohammed Yassin Mustafa, Dr. Sara Mustafa Radwan. Alhamdulillah, we published a review article on the topic in a journal called the Schmeidberg's Archives of Pharmacology, and a research article was published recently in International Journal of Molecular Sciences, while our based pan-cancer analysis paper is still under review. We also presented that work in the 11th Conference of Human Genetics and Genome Research Institute during October 2025 and at ASU International Clinical Oncology Conference in December 2025. Here's our table of content, starting with introduction. In Egypt, HCC remains mainly caused by HCV and to a lesser extent by HPV, remains the most common cancer and the leading cause of cancer death, responsible for 1/3 of all cancer fatalities. It's a complex disease and with limited treatment options, and most of them are only effective at an early stage, while at advanced stage, this cancer is associated with poor prognosis due to frequent cancer metastasis, tumor recurrence, and lack of curative treatment. Tissue biopsies aren't routinely conducted. Liquid biopsies still show some promise. So the need for early detection, non-invasive liquid biopsy markers, especially in metastasis, is of a great need. Epigenetic derangements, one of the cancer hallmarks regarding long non-coding RNAs. Long non-coding RNAs are involved in cell proliferation, differentiation, and metabolism. They are dysregulated in different cancers, having critical roles in various post-transcriptional processes. Different reports implied several long non-coding RNAs' role in the progression and metastasis of HCC, such as our main target here, HOTTIP or HOXA transcript at the distal tip. Dysregulation of HOTTIP is associated with various malignancies, including HCC. HOTTIP promotes proliferation metastasis and has oncogenic and metabolic reprogramming role. One of the most common downstream effector of HOTTIP is a protein called WDR5 or WD repeat domain 5. Single nucleotide polymorphisms or SNPs. When SNPs are inherited together in regions with high linkage disequilibrium, they are known as haplotypes. SNPs and their haplotypes can alter the expression of the coding genes and the protein non-coding genes like long non-coding RNAs. Specific HOTTIP SNPs have the potential to be biomarkers for HCC risk and prognosis. One haplotype, RS175, RS206, RS174, showed a 1.91-fold increased risk of HCC as stated by Wang et al. This study was performed in Chinese patient samples. So the problem is that in Egypt, HCC represents the most common cancer. we hypothesized that HOTTIP haplotype RS175 RS206 differentiates metastatic from non-metastatic HCC and subsequently affects prognosis and survival in HCC patients. Furthermore, we hypothesized that metabolic derangements in metastatic liver cancer will be echoed in circulating blood, which may enable early blood-based non-invasive diagnostic testing. For the subjects and methods, we recruited 198 patients from Ain Shams University hospitals, divided into two groups. Group 1 included 129 non-metastatic HCC patients. Group 2 included 69 metastatic HCC patients. They were adults of either gender, diagnosed by alpha-fetoprotein and CT scan, socioeconomic, age, residence matched as much as possible. The exclusion criteria included any patient with liver transplant, other primary cancers, renal insufficiency, thyroid dysfunction, incomplete patient record. The sample size calculation was based on Quanto software. We collected the patient samples from data sheets, including histopathology, lab tests, imaging, survival data. We withdrew blood samples for DNA extraction and genotyping, SNP assay analysis using real-time PCR. We performed also routine blood sample tests, including CBC, liver function tests, kidney function tests, tumor markers such as CEA, CA19.9, alpha-fetoprotein. For the molecular docking study, we performed long non-coding RNA target analysis to target the downstream target of HOTTIP, which is WDR5. For such, we designed short interfering RNAs disrupting that interaction between both long non-coding RNA and downstream target. For statistical analysis, we used SPSS and MidCalc. For survival analysis, root curve analysis was performed, and odds ratio, hazard ratio, area under the curve was calculated. For the results section, they are subdivided into 6 sections, starting with association with the risk of metastasis. This is the first study to investigate the myeloid allele frequency MAF and genotype distribution of the HOTTIP gene haplotypes RS175, RS206, and its association with metastasis in Egyptian population of patients with HCC, as represented in that table. The TT and TG genotypes of the codominant genetic model was associated with a significantly decreased risk of HCC compared with individuals carrying the GG genotype. The dominant genetic model of the same SNP showed a significant 2.25-fold increase in the odds of metastasis. Under the recessive genetic model, we failed to find a significant difference in the odds of metastasis. Additionally, in terms of alleles, a significantly decreased risk was observed in the T allele compared to the G allele. In terms of the second SNP, RS206, the GG and GC genotypes of the codominant genetic model were associated with a significantly decreased risk of metastasis compared with individuals carrying the CC genotype. The dominant genetic model showed a significantly decrease in the odds of metastasis. Under the recessive genetic model, we failed to find a significant difference in the odds of metastasis. Additionally, a significantly decreased borderline risk was observed in the G allele compared to the C allele. This finding was in agreement with the Hardy-Weinberg equilibrium, with a p-value of higher than 0.05 in the whole population and the individual study groups. Additionally, we analyzed the relationship between H long non-coding RNA HOTTIP and SNP and HCC metastasis by Cox regression model by using groups as covariates. We found that RS175 was associated with a significantly lower risk of metastasis in the TG and TT genotypes or subgroups than in the GG subgroup. However, only the CG subgroup of RS206 had a significantly lower risk of metastasis. For the prognosis, association of HOTTIP SNPs with disease prognosis, ROC curves for evaluating the prognostic value of different variants reveal significantly moderate association with prognosis, so it cannot be used as standalone for predicting prognosis of the patients. In terms of diagnosis, ROC curve was used to discriminate between HCC metastatic and non-metastatic groups and for evaluating the diagnostic value of different variants. This revealed also a significant moderate to good association with diagnosis. In terms of patient survival, the metastatic group had a significantly lower overall survival than did the non-metastatic group. Kaplan-Meier survival analysis revealed that the survival time of the metastatic group was significantly lower than that of the non-metastatic group. Moreover, the cumulative survival function for both patient groups, determined by the hazard function, confirmed that the metastatic group had significantly lower survival. In addition, the GG genotype and TG genotype for RS175 had significantly lower survival time estimates than did the TT genotype, whereas the CC and GG genotypes for RS206 had a significantly lower survival time estimates than did the CG genotype. In terms of allele effects, the G allele had a significantly lower survival time estimates than did the T allele, whereas the C allele of RS206 had a significantly lower survival time than did the G allele. Furthermore, we studied the impact of HOTTIP polymorphism on overall survival with adjusting Cox model for confounders. We took these factors in consideration, including age, gender, treatment, etiology, HPV versus HCC as confounders. We found that RS175 is a major survival determinant, including for the TG subgroup shows high risk, GG subgroup is protective in metastasis. For RS206, it has limited value, and we found that metastatic status is a dominant predictor for survival. Joint effect and results of haplotype analysis compared with those in the metastatic group, the combined impact of the investigated gene polymorphisms in patients with non-metastatic HCC was investigated. The findings revealed that the GC haplotype was the only haplotype that protected against HCC metastasis. Furthermore, the combined heterozygosity TT plus CC versus TT plus GG was a lower risk factor for HCC metastasis. This was also applied for the heterozygosity, the combined heterozygosity TG plus CC versus TT plus CC. For the molecular docking results, the target with the best scores were chosen where there's 3D structure, energy, and hybridization energy were tabulated. Additionally, we verified that these short interfering RNAs are the best ones by ensuring that they can inhibit the direct interaction between HOTTIP and WDR5. So in conclusion, as a take-home message, this is the first study to investigate MAF or minor allele frequency, genotype distribution, and the prognosis and survival related to HOTTIP SNPs in HCC Egyptian patients. We found differing allele frequencies and genotypes compared to African and global population, likely due to unique genetic ancestry. We found some protective genotypes for RS175, TT, and TG genotypes linked to reduced metastatic risk, while for RS206, GG, and GC genotypes also showed reduced metastatic risk. For haplotype analysis, we identified protective haplotypes, GG versus TG, TT plus CC versus TT plus GG, TG plus CC versus TT plus GG, which were associated with decreased metastatic risk. For survival outcomes, better prognosis linked with TT RS175 and GC RS206 genotypes. For diagnostic and prognostic value ROC curve analysis, confirmed limited predictive power of some genetic models. For the therapeutic innovation, molecular docking identified short interfering RNAs targeting HOTTIP WDR5 interaction. For the future potential, short interfering RNA delivery systems and epigenetic targeted therapies warrant further exploration. Here's a graphical abstract summarizing our findings created by Render. Thank you. Do you have any questions?
Thank you so much, Mona, for your lovely talk. We do have some questions for you. So will your data on the GC genotype specifically permit change in treatment to maybe more aggressive treatment and maybe beyond the siRNAs to other potential approaches?
Sorry, again, ma'am. Sorry.
Yeah, so will your data on the GC genotype permit change in treatment potentially to a more aggressive form of treatment, or maybe beyond siRNA to other treatments as well?
So it's a very nice question. I think this is still a theoretical assumption. We only found in our patients that this will be protective, associated with better prognosis, allows them to have a longer overall survival, and so on. We can't say hypothetically that this will lead to more aggressive treatment or to lesser extent a less aggressive treatment or so. So I think this question cannot be answered directly as I didn't perform such tests.
Got it. Yeah,
This
thank you so
will
much.
further experiments, I think.
Yeah, no, it's a future route, so it's lots to explore. Another question that we had was the patients that you were discussing were stratified based on socioeconomic factors. Do you see any correlation between the SNPs with any of these factors?
Yes, we performed a modified Cox regression model to exclude factors, some clinical factors such as age, gender, treatment received by the patients, either they received treatment or not, the etiology leading to HCC like HCV, HPV. In Egypt, we have still a little bit much HCV patients. We excluded all that factors and found that the only confounder that affects significantly the survival was the age, while other confounders didn't affect the results of our study.
Interesting. There was a question kind of leading out of what you were just talking about in terms of, specificity of the genes. So this hot tip gene is dominant amongst which population of the Egyptian population specifically?
Um, we mentioned in the presentation the distribution of each, wait a minute. So we mentioned the distribution of the HOTTIP among our population. Um, I can't state how or how much is the percentage among all the Egyptian population, but among my studied group, which includes 198 patients, uh, we found that 100, wait a minute, I need to give you a specific number. We found 76% was T allele, while 23% was the G allele, or the non-metastatic group. 46% was carrying the T allele, while 35% was carrying the G allele. This is among our studied group, not the whole population. I think no one can take or tell that information. It needs screening the whole population.
Yes, for sure. A lot of work, a lot of work. Another question, in terms of delivery systems, is there potential for use of things like nanoparticles or any other sort of delivery system?
Yes, this is a good question. It's part of our future perspective to take a step, not only saying it's okay, we have designed short interfering RNA. We want to help really patients with HCC with a really treatment. So it's part of future perspective, but needs a more fund, you know, needs a lot of fund actually to
Yes, for
produce
sure.
it.
And in terms of also delivery mechanisms, have you looked into potential like toxicity that's related with using these like siRNAs therapeutically because there is like a developmental like gene regulation aspect to it. So could there be potential toxicities associated with it that need to be considered before bringing these into grand scale usage in
Yeah,
therapy?
that's right. We need to test this short interfering RNAs in vitro and in vivo before being upgraded to clinical trial. This will take very huge steps.
Yes, for sure. There was also a question asking whether you can clarify if you've done any preliminary data evaluating the prognosis in biomarker patients in other related diseases as well.
We have We have divided our group of patients into poor prognosis and good prognosis based on what a score called API score. This score mentions the cirrhosis, fibrosis status of the patient at the stage of admission. So at certain, a cutoff value was determined via the ROC curve analysis, and it was 0.5. The patients having API score higher than zero point 0.5 will have a poor prognosis, while patients having API score lower than zero point 0.5 will have good prognosis. Is that the answer of the question, or I didn't clearly hear?
Potentially, maybe, yeah, it was more of a clarification. So yeah, maybe it was just misunderstanding. But another question was whether this target gene have any association with patient heredity as well. I'm assuming
Sorry,
yes.
sorry, the connection was interrupted. I couldn't
Yeah,
hear
if
you.
there's a
Sorry, what
connect, so sorry. If the target gene has any association with patient heredity.
was patient what? Sorry.
Heredity, so like, like genes being passed on, right? Like if
Yeah, yes. Although it's a gene in the non-coding coding for long non-coding RNA, it still can be passed from one generation to the other.
yeah, that would make sense. That would make a lot of sense. Yeah, thank you so much for answering the questions, for the amazing
Thank you.
presentation. We really
Thank you,
appreciate your
Irina.
work and your time. Um,
Thank you, Irina.